So thanks for inviting me, and it is just coming back my second home, and I am really happy to be here as well as to see our next generation. They are so much interested in science. So before starting my actual talk, okay, let me ask a small question to my friends in the audience from school. So when do we become scientist? Do we need any formal training? Just one slide on that. Okay. So if you ask yourself, how do we learn? Okay, what are the sources from where we learn? So this is my personal take. I don't think that it is very unique. Some of the learning process happens from book. Okay, we read book, we learn something. We also learn from our teachers, definitely. We also learn from our friends. And there are another way we can learn to teach ourselves. Okay, this is basically a way which I see that these four ways I can learn. Okay, one of the important component which we generally forget, that teach yourself. Okay, now say if we place ourselves from primary school to high school to college to higher education, what are the percentage of this learning process changes? Okay, this is kind of coordinate. Left hand side you assume it is a primary stage, and right hand side pie chart basically you think it is a advanced beyond college and so on and so forth. Just as you assume on that. Now in the primary stage you can see that the teacher's role is maximum. Okay, the blue color. Okay, definitely you read books and you learn something from your friends. When you become much older, okay, from primary school to high school to colleges, the role of teacher decreases. Okay, and definitely the component which you learn from books are increases. When you go further, there is another component, it start appearing. It's the teaching yourself. Okay. Now, once the role of teachers goes down, role of books goes down, roles of friends goes down, but teaching yourself become more prominent by the time you become scientist. So job of the scientist, you teach yourself. Nobody is there to teach you. So once you know that I am learning through myself, asking questions, looking for answer, there is no source of answers of my question which I am asking. I am finding out the answer. By that time you become scientist. So you start asking question yourself which answer is not known, and you find out the answer of that question. If you find out the answer, you become scientist. That's the way I define to become a scientist. It doesn't have any age. So coming back to my main topic, this is a kind of hypothetical park, zoological park. Okay, it doesn't exist so many, animals in a single place. Okay, hypothetical. Now, if I ask a fundamental question, what are the living beings in this image? Okay. Yeah, animals, then plants, then water. Okay, then microorganism you cannot see. Okay, good. So what are those microorganisms? Do you know? Bacteria, virus, algae, fungi. Okay. So how do we define to tell it is a living organism? No, bacteria also living organism, I am telling. The tiger also living organism, I myself also living organism. How do I define that a particular system is a living organism? So if I stop myself, I do not move, I am not living organism anymore. Huh? Sorry? Metabolism. Oh, many things. But always metabolism doesn't happen. For all living beings, metabolism doesn't happen. All living beings doesn't move. Okay, I'm not asking you the knowledge from book. You ask yourself, how do I define a living? Okay, let me answer it. Which can replicate? Which can double? Which can multiply? Which can propagate? Okay, can be defined in a layman term as a living organism. Am I wrong? Not really, but I am not 100% also correct, but I am not wrong. Okay. Now what makes to propagate? How do I know that it is going to propagate? Okay. There is a system or material, so we call it genetic material. Okay. Genetic material you have read in your textbook, DNA. Most of you have read, no? DNA. At least you have heard RNA. These are the genetic materials. Okay. That actually helps to replicate. That could be for virus, bacteria, human, tiger, anything. Okay. Unless until a certain system doesn't have genetic material, generally we do not call it a living organism. Okay. Wherever you see that there is a genetic material, I can define that could be a living organism. Okay. That's the definition. Now, what is this genetic material? Okay. Let's say eukaryotic, like, we have so many cells in our body. It's a simplistic representation. It is a cell, human cell, you can assume. Within cell, there are some compartment. Even if you do not know their nomenclature, doesn't matter. Okay. Within that, there is a storage of genetic material. Okay. This architecture varies from organism to organism, from microorganism to, say, human being. It's not always correct that I have nucleus, within that I have genetic material. But the important thing that each and every living organism has genetic material. Okay, DNA or RNA, most of the cases DNA, some of the cases RNA. What is DNA? It is a double-stranded kind of system, and there are 4 letters generally. Do you know that? Most of you know. A, T, adenine, thymine, cytosine, guanosine. Okay? And, there is a complementarity. Okay? Adenine will interact with thymine and guanine with cytosine. These are the textbook most of you have read. And this is basically complementary. If it is adenine, the opposite strand is thymine. Okay. Now this basically genetic material. When I talk about human genetic material, I have written here. If you extend it, like like a CC carbon bond, you extend it. It's a very long distance. Very layman term. You can 150,000 times you can go moon and come back. It's so long. Okay, which means that it's a very, very, long information are stored in these genetic materials. Okay, like a novel. And in a novel, that each and every sentence is not important. Okay, when say I have read a novel and I am explaining you, maybe a 200-page novel, I can explain you within 15 minutes. That means, some information which is important to define that novel. Okay, you don't need that 200 pages of text. Okay, sometimes it is important, sometimes it's not important. Just create an environment to represent this novel. In genetic material, we also have similar kind of, it's not that all part of genetic materials, AT, GC, everything is important. But there are certain portion in that material or in that long text, there are very, very important sentences are there which cannot be taken in a wrong context. Okay. Now, in that important region, even a letter change can create a havoc problem. Okay, that's the important. Okay, it is not that anywhere this change makes create problem. No, there are certain important region where even a single base or even a single letter change can cause a huge problem. One of the problem in our country is sickle cell anemia. It doesn't happen most of the population, but tribal population which is very high density in Odisha, Chhattisgarh, Madhya Pradesh and Gujarat part, tribal population, just one base change can destroy the ability to carry oxygen by hemoglobin. Most of you know hemoglobin, what it helps to carry the oxygen. Just only one letter change makes that hemoglobin inefficient to carry oxygen. As a result, the normal blood cell get coagulate or aggregates, do not carry oxygen and get deposited within the vein and a crisis creates. Okay. Why it creates? Most of you, again, it is a textbook knowledge. DNA code for RNA, RNA code for protein. And one amino acids in a protein string is coded by 3 base. Okay, this is again textbook. So this amino acid will get changed. Even you don't need to understand that, what is changed and how it is going to change. The important message is there, one letter change can create a problem or create a disease. Now, sickle cell anemia is one of such problem. I am not going to discuss. Our objective is to how to correct this in Indian population so that we do not have burden of sickle cell. That is the research we are doing in our lab. So this is an example. Okay, you have come Indian Institute of Chemical Technology. It is established in 1944, and there is a sentence. Okay, what happens if a letter changes? It changes the meaning. You do not get any meaning out of it. It's an important sentence, and one letter kills the meaning of this sentence. Now, question is that few years back, can I correct this sentence? Okay, when you type in a computer, you have done mistake. You can delete it and rewrite it. Can I invent biological tool which can read this sentence and correct wherever the mistakes is there? That the question was in few years back. Like, can I correct this letter, one base change, one letter change using some biological tools or biological means? Yes, we can. In 2012, 2 lady professors, one from University of California, Berkeley, another from she is Jennifer Doudna and Charpentier from Paris. They collaborated to invent this tool and published one of the outstanding journal in science, which is Science. It's known very top-notch journal of our scientific community. In 2012, they have published and showed that bacteria has a system which can be reprogrammed or re-engineered, okay, such a way that it can go to any kind of genome and correct it. That's the invention they have made in 2012. As a result, in 2020, they got Nobel Prize in chemistry. Okay, it's not that old technology. It is just discovered in 2012, started practicing in the year of 2015-16, and very soon they got the Nobel Prize having the potential to correct any disease which caused by genetic changes. Okay, it could be for human, it could be for any animal, it could be for plant, and so on and so forth. Huge implications. Okay, and one of the most expensive patent at this moment in the world. Today morning, Dr. Bedajyoti explained what is IP patent. They hold the patent. Okay, now they are filthy rich. What it does basically, as I told you, there is a mistake, okay, and I need a machine, tools. Okay, and that tools cannot be made only one component. Like say your cell phone, it does have many things. It has speaker, it has microphone, it has processor, everything. Okay. Similar way, when you design any biological tools or biology design any tools for their own function, it has many components. In this case, there are two components. One is a small piece of RNA. Okay, nucleic acid. Most of you told complementary, like A bind to T, G bind to C, and so and so forth, and a protein. Okay, small protein. Protein are functional most of the cases. Like when I am talking to you, okay, there are some proteins are involved. How loudly I can talk, how politely I can talk, everything has been decided. That function is going to be decided. The nature of the protein, the amount of the protein, and so on and so forth. Whatever we are doing, okay, these are mostly functional materials for our body. So this is the protein, but this protein are from bacteria, because bacteria has used this technology for defense mechanism. Okay, it is not practiced in our body, but these two scientists, they have reprogrammed so that it can work in our body also, or any body like animals, any animals or plant. So it has two components, and they make partner to each other like a complex. In literature, it is known as a ribonucleic acid protein complex, in short RNP complex. Now this complex, when we introduce to any genetic material, depending on the design and certain characteristics in this protein, they will find out exactly position where you want to make change. Like address, when you are sending a letter, if you do not write any address and post it any post box, it will not go anywhere, no? So you need an address. So these two complex, those two material, when they form complex, they decide the address. Now this address you can design putting some kind of basis here so that it can go wherever you want. It's a programmed design. Now when you design and ask that to come and sit here, this protein, it has an inherent ability to make a change, like chemistry. Okay, say if you have hydrochloric acid in one bottle and say NaOH, another bottle, you mix them, they will react. Okay, same way, when they come and sit here, they can do certain kind of changes, because all this molecule or ATGC base, it's a chemical molecules. Okay, it has functional group, it can go for chemical transformation. If you give a proper reagents through this kind of complex, it can change whatever you want to bring change. Now the advantage why I am doing so much complex system, because I do not want to create change anywhere else. Okay, I want to create in a particular positions that change, because that is the change in the later is creating problem. Okay, if I make change anywhere else, I will introduce another problem. Okay, rather I am correcting, I am introducing another problem. So I do not want to make a change anywhere else where there is no mistake. I want to make a change where the mistakes is there, so that it can go back its actual information. But as I told you, it's any genetic material, it's a very long text. There are many sentences, many paragraph, many pages kind of stuff. So similar kind of phrase or continuous word may be available here and there. Okay. In this case, like say, almost same context. The Indian Institute of Science is a public and deemed blah, 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 blah. You can see that there are some similarities. Okay. And it is there in the same novel, same genetic material. Now, if you have similar kind of sentence, it can go and make the changes such a way that, okay, it's introduce a problem. Okay. So you wanted to correct one sentence, but as a result, you have created a problem for another sentence. Okay. That is not desirable, because we are talking about therapeutics. We are talking about change which can reverse the disease. Okay. If you make a change in an important region of that text, then again you are losing a meaning of somewhere else, which means that it can create a disease for something else. It is not tolerable. So there are two things we have to remember. One is efficacy. We know efficacy, like say you have a small auto versus a truck. Definitely truck will carry more load, so efficacy is more. That's called efficacy. How big, how much, how many, how better is the efficacy. Okay. Another term, it's a specificity, in the sense that if you have a truck and a railway, truck will carry some kind of goods, railway carry passenger, or aeroplane will carry passenger. Okay. Vice versa may not happen. That's what we call specificity. Okay. The intent, it has been made for certain purpose. If it is doing the only that purpose, we call it a highly specific. If it is doing something else along with that, we call it has very low specificity. So you have to have very high efficient and high specific, system. Now, as I told you, this is a protein. It has a ability to, create change in molecules. In laboratory, what we can do, so if you assume this is the portion which basically makes the changes or create the changes, I can design such a way, or I can engineer such a way that this ability goes out. Okay. It cannot change anymore. It cannot change anymore. That doesn't mean that it will not bind with its partner, that RNA. It will bind still. Not only that, it will go and sit wherever you want. Only thing is that there is a mistake, it will not be able to correct the mistake. Rest of the job will be done. Which indicates that if I have a sentence in a novel where I cannot go each and every part of the novel and I find out whether this sentence exists in this novel or not. If this is the question, I can design this system and through this novel, it will automatically go and see it where I want to see it and tell that yes, this sentence is available here. Okay. Now if I know only one novel has this sentence, no other novel has this sentence, I can tell just without knowing that what is the novel, I can tell if this sentence exists, that must be this novel. Okay. That means this can help us to identify genetic material of our choice. Say, if you ask a question, I have a series of test tube. I have human DNA, I have say cow DNA, goat DNA, fish DNA and all, but nothing is labeled. And I want to ask, out of this 10 test tube, is there any test tube which contains human genome? If this is the question, then I will design a CRISPR RNP complex such a way that it can find a unique sentence which is only available in human genome. Okay, there must be, because I am much more different than fish. There should be some unique which will define me. Okay, I have to find out that sentence which is known because I know human genome. And I can design this to dictate or to identify out of this certain, test tube, is there any test tube which contain human genome or not? Okay, that's fine. It will not make change, but it will go and sit on that sentence. Now, most of you told microbes are there. Microbes I cannot see. Many microbes are there everywhere. Even if you scratch your hand right now, there will be maybe 10, 20 different type of bacteria in that scratch you have got. But all microbes are not dangerous. Some microbes are dangerous. That most of you have heard 3 years, 4 years back, coronavirus. It's a dangerous. Okay. And the question was that, can I detect coronavirus? Okay, there are a lot of technologies are available. And one of the technology you have heard from your parents, RT-PCR, do RT-PCR. Okay, when each and every individual almost were getting infected, okay, the availability of that kit was very low. Okay, and also was not, because we do not produce in our country RT-PCR kit at that time. Now we produce a lot, but in 2020 March, we didn't have any single company who used to produce that kit. Okay, so that means we can produce a system which can detect coronavirus, but that was not our objective. We were working for something else. Okay, but logically we can create. So I have so many microbes. You assume one of the microbes is very dangerous, which is corona. Okay, I take a sample from your body fluid, wherever. Okay, it was swab, because it has been understood that swab contains maximum number of viral particle of corona. That's why we used to swab. That doesn't mean that it doesn't available in any other, but number of particles are less. Or number of virus is less. That's why I need something substantial amount to decide. So many microbes are there, all are not bad, only one is bad, and I need to find out that bad microbes is there in your sample or not. That's the question we used to ask. Okay, for each and every microbes detections. That is a unique sentence. Now to make it simple, say you have coronavirus with something else. This is not the situation only you have two, we have many. But to make you understand, I have only taken 2 examples. Now, as I told you, the text of these microbes and text of this microbe are different. There are some text will be unique for them. There will be some text unique for them. But I want to know whether a particular microbe is there or not in that system. So this text is corresponding to this, and this text is corresponding to this. And I want to know that whether this microbes is present or not. So I will design this complex to read this sentence, not this sentence. Okay. So that's what we do. We have designed, but the main problem is that the amount of, say you have only 1 novel or 2 novel. Okay. If you read only one same novel of 2 copies, okay, by 2 different people, may not be easy, faster to identify very easily. So you need number of sentence more. There is a technology we can amplify like a Xerox. You have a 1 page, you go to a Xerox machine, put it, 100 copies you can produce of the same text. Same way in biology, there is a technique called PCR, polymer chain reactions. Okay. Again, you don't need to go in detail how it is does, what it does, but what it principally do that same sentence it can amplify. Okay. Number of sentence can be increased. That's what we do. But this PCR technology is not foolproof, which means that when I have genetic material from other component, it amplifies certain amount. Okay, like if it is getting amplified 10 times, it will amplify 2 times, but will be amplified. It's not that specific. But challenge is that can I read the actual sentence which I am looking for? Okay, that means I will design a CRISPR complex system to read this sentence because I am looking for a particular virus which has this sentence. So I do that and they sit, they do not sit here. So far is good. But if I want to read it or see it, this cannot be seen. In the sense, giving an example, say, as I told you, I scratch my skin and put it in a test tube, some solution, there will be some microbes, but I cannot see that. And within this microbes, there are the sentences. Again, I cannot see. I have to create a system where I can visually see. Like, there should be color, something colorful. Again, as I told you, we can do engineering. What do we do? The protein which we have, this Cas9 protein, we can tag with any kind of fluorescent molecule, or which can give you color. That's the we call fluorescent molecule. Or could be a very recent time, there is a system called quantum dots, you might have heard, which is very bright color. Even very low concentration give you very, very bright color. So quantum dots are nanoparticles. So we, in lab, we can design protein and connect this kind of fluorophore very easily. Okay, there are biological chemistry is available which you can tag. Now this protein become colorful. Okay, you can see by naked eye. Now if you allow to make a complex, complex acts as become color. Okay. Now we can see it individually. If you put in test tube, if this you are tagged with a red color fluorophore, the solution become red. That's the way we see. Now you put them in a genetic material. Along with that, they will go and sit there. Now all this sentence become colorful. Okay. If you have a genetic material in a test tube and you are looking for a particular genetic material, you design this system to read that particular genetic material with a labeled complex, and if this genetic material is available in this test tube, it will go and bind and make that positions very colorful. Okay, that's what we make it colorful. Now what do we do next? With this principle, we have made a team in 2006, 17, 18 onwards, we call Feluda team, because the assay named is FN Cas9 editor linked uniform detection assay. Okay. Now it has two meaning. Definitely it gives a indications what kind of systems we are using. Second meaning is that both the scientists who are working for this problem are Bengali. Okay. And there is a novel, okay, whose hero is basically, it is a series of detective novels written by Satyajit Ray. Those who are Bengali might have read so many books of Satyajit Ray. So one of the series is Feluda series. Okay, and there is a detective who solves each and every kind of detective problem. Okay, so this is the best way to give the name of our kit, because we are going to detect something which is not easy to solve. So this is the team, 3 students and 2 scientists. We joined together, we made a separate team. We have to have some kind of detection system for nucleic acid identification. That time coronavirus was not there. So how we used to do test? As I told you, this is the sentence you assume. We can amplify, as I told you, polymer chain reaction. During amplification, there is a mechanism we can introduce this kind of arrow. I will explain why I have used this arrow. Okay, this is a kind of modification. Okay, it's like you can assume this is a iron ball. Okay, now you put a magnetic strip here and put iron ball to roll. Okay, now when it is rolling and crossing the magnetic strip on the floor, what will happen? On the magnetic bead, on the magnetic magnetic plate, all the balls will come and stuck because it is a magnet, no? Assume that. Okay. So this is kind of iron ball and has a tendency to stuck on any magnet. Okay. Now, once I have amplified with that magnetic ball, I have a CRISPR complex which is labeled and I mix them. As I told you, when I am amplifying, there will be some sentence which I want to read that will amplify. There are some sentence I do not know, I do not want to read, they also will amplify in a low quantity. But this complex will go and label only the sentence which I want to read. But there are certain sentence which I do not want to read. Some other microbes, it is coming from some other microbes. Assume this is coming from corona, this is coming from say E. coli. We have many E. coli in our body, still doesn't have any problem. Okay. Now there is a techniques called lateral flow assay. This is well known. Okay. You take this solution and put it one part of this, this is basically nitrocellulose membrane. Okay. And in the membrane there is a coating we call streptavidin coating, but for you it is kind of the magnet we have placed it. Okay. In biology it is a biotin and this is streptavidin. Biotin, streptavidin, whenever they see each other, they stuck to each other very easily. Okay. But you don't need to remember biotin, streptavidin. What do you remember? There is a kind of iron ball and I have magnetic strip here. Now you put this solution here, capillary action, like we put a drop of water on a newspaper, they spread, they spread due to some action, we call capillary action. Same way, you put here, this solution will move this direction due to capillary action. Let's see, it is moving. Okay. When it is moving, this complex will pass through this magnetic strip. If it is passing through magnetic strip, as these are kind of, iron ball, they will stick here. That means they will get deposited in a small region and the intensity of the color will increase. Now you assume you have only yellow color, no green color was there. So only yellow color will get deposited. If yellow color is getting deposited, you don't see any color development because that the complex we have made labeled, they do not make any partner with the yellow because we have not intended to read that yellow portion. Okay, so there is no color. You assume there are certain portion of the green which I wanted to read, or I know that sentence belongs to coronavirus. So if this is there, some of the portion will be labeled and will get deposited here. If that is getting deposited, what you will see? There is a color development, red color. Okay. So red color in this region will indicate that the sentence you are looking for is there or not in an original sample. If you get yes, sentence is there, the sample where from you are collected, you will tell that yes, this is the novel is there. That means if you are asking a particular region of coronavirus, you are going to read it. If the color has got developed, the sample you have collected must be containing the coronavirus. So having color and no having color will tell whether the coronavirus present or absent. Not only that, the amount also, the intensity of color will tell how many this blue color you have started. Because if you have a one copy, you want to make 10 copies, okay, one copy will give 10 copies. But if you start with two copies, you will end up with 20 copies. It is not that linearly correlated, but proportionately correlated. That means the intensity of this region will indicate what is the original quantity you have started with. That will tell you the viral load or bacterial load, what you are looking for. So that's the basically system we developed for other region to detect the mutation change in the sickle cell anemia. We wanted to correct, but when we visited, we used to visit a lot of this tribal population area, we realized that we need a test also. That was the motivation in 2017-18. The current procedure is that you take small amount of blood by punching on a finger of a small kid and dissolve it in a particular solution. The blood get precipitated. That will indicate you have sickle cell anemia diseased or not. But this process has two problems. One is sensitivity. When like you want to test 100, you may miss 20 times. So 20% will not show positive in this kind of test. Not only that, when you are punching a small kid, 4 years, 5 years, 6 years, it's a painful for them, and they do not want to come and volunteer and get screened. It's a very painful. If you go across this kind of procedure, you will see this test is not going to work too much. So we replace with this test by our test, where you can just collect. Our saliva contains DNA. What I need genetic material, nothing else. This, saliva will contain our DNA. I can collect from saliva the DNA. I can do the test which I have explained right now. Okay. Now I can ask, to queue, for a queue, give saliva, do not give blood. We will take blood only for, your parents. Okay. It's very successful. We, students are happy, comes, what is happening. Everybody, always excited to something new. Okay. And this test become very, very, popular in terms of acceptability. Right now, still we have not gone to the commercial level that it can be available in the market. Anybody can go and buy and test it. We have not achieved. But when do ourselves in a field trip, we see that there is a hope of success. So this test, basically, we got converted for corona testing when it was demanded in 2020 March onwards. So, we within a month we have, converted this sickle cell anemia screening test to the corona testing, and that's the basically news most of you have heard and read. But the success happened when, this was bought by Tata Sons initially, then they have opened a new company, Tata MD, Medical and Diagnostics. You can go through their website. That is, they have very, proudly claim that this is the first product for the Tata MD, and it was very much successful in the mobile van. Most of you know, the going to clinic was a challenge, and in the village, clinics were not available. So you have to put clinics on a mobile van and take to the village. But when this sophisticated machine was placed to a mobile van, okay, after certain journey, they do not function anymore. But as our test doesn't need very sophisticated instrument, they are very, very successful on mobile van, as well as some places like Dehradun, Manipur. Okay, this is tweet by Manipur, chief minister, that they were really, really extremely happy to have Feluda in their region, which has solved a certain fraction of their problem. I will not tell the entire problem, but certain fraction. With this, I will end here. I hope I am able to communicate what we have intended to do. With that, thanks to my, colleague, friend, collaborator, Devajyoti Chakraborty. He is a biologist, I am a chemist, and most of the students, for kids, this, photo, we do not do always science in the lab. We also have live. We go to, restaurant, we go to, excursion, field trip, it's very fun. Okay. With this, again, thanks to alumni, Jadavpur alumni, to give me opportunity to speak to you, and definitely I will be happy to talk to you if you have any doubts. Thank you very much.
Thank you so much, Dr. Maiti, for explaining this in a wonderfully lucid manner for the young children to understand. I now request our president and the senior. Okay, sure. Any questions? We can facilitate that.
If you want to ask any question to Dr. Souvik Maiti, you can ask. I can go with my mic. Yeah, anyone, please. He's a great scientist of India, Dr. Bhatnagar Award winner, which is considered as the Nobel Prize of India. Yes, ma'am. Your name?
Varsha. I'm Varsha Arun from MDAV Public School, and a few days before I was studying on CRISPR, and my main question is, we got to know that Feluda was used, and we got to know all the pictures on how it was used to detect COVID-19, but was this really a success? And we would like to know its journey past after it was discovered.
Yeah, great question. Yeah, this is the question always we face, being an inventor of this Feluda. The success has two steps. One is the invention, and second is the application. Okay, application part is basically based on lots of dynamics, depending on how scale up is happening, the business hub, how they have penetration, how good their sales, lot of other competitions. Okay. Now, if you measure the success in terms of the number of selling kits at that time in our country, I will tell no. Absolutely no. We have sold, according to the market, according to the Tata MD, the number they have given is few lakhs. Okay, it's not even 35 to 40 lakhs. Okay, has been sold. Other than there are certain activities which call corporate society activities, like in a van you see, it has gone free of cost. The Manipur, activities you have seen, it is a free of cost. So, there is no commercial angle. Other than that, the kit has been sold by price only 35 to 40 lakhs. That's the number we know, because the royalty we are getting up for that only. Okay, if they have sold more than that, we do not know. So if you ask very precisely, is it successful when we have sold 10 crore or 20 crores of RT-PCR kit? Out of 20 crores, only say 50 lakhs has been sold Feluda kit. So it's absolutely not successful. But if you go back and ask that whether technology is successful or not, I will tell yes. If I can detect 35 lakhs, give me opportunity, make 5 different factories, I can make it 35 crore. It's matter of scaling up. Okay, matter of availability. When you use the scaling up from 1 kit to 1,000 kit, the price also goes down. Okay, that also decide how many you are going to sell in the market. Okay, so as a technology, it is successful. As a business model, it is not successful. So that's the honest answer I can give you. Not only that, just small information I want to give you as a scientific. The RT-PCR kit, which most of us we have used, okay, is a 40 years old technology. Okay, but whereas CRISPR technology, as you seen from my lecture, it is a Nobel Prize has been given in 2020. It's a 3 years old kit. Okay, old technology. You can take an example like a smartphone. Nowadays everybody uses smartphone. Even a sabji wala in your vicinity, he is also using smartphone, paying Paytm, etc., etc. When smartphone came 10 years back, if I give smartphone to a sabji wala, do you think he could have used as comfortable as he is using now? No. Okay, CRISPR is at that stage. 10 years back, the smartphone was in our society. Okay, if you try to use more and more, okay, each and every clinic also will know, okay, this is the way it has to be done. Okay, the knowledge of use also important to make this technology successful. That knowledge is very limited because it is a new technology. So down the line, maybe 10 years down the line, it could be like, there will not be RT-PCR kit, there will be only CRISPR based kit. If you go literature, you will see all diagnostics will be replaced by CRISPR-based kit because it's very specific. Okay,
Yeah.
it's highly specific.
Dr. Souvik, I want to give opportunity to
Yeah.
a few students. One, I think two seniors want to ask also question, but please, I want to give the opportunity to student first.
Thank you, sir. So, if we have, we just contracted disease corona, and we have about 2 samples of DNA in the sample we have taken. So, is it possible that the amplification phase fails and we only get very less amount of that? And then will we be able to identify if we have contracted disease or not?
Yeah, if you do not have good amplification step, okay, you will fail. You will fail to detect. Okay, because all these techniques, okay, real-time PCR or CRISPR or anything else, you need amplification step. That's why they are very, gold standard kit. Okay. Now, what is the alternative? Alternative is basically either do the test or some other methods. But there is no other alternative methods to detect so precisely when your amplification do not work.
Dr. Souvik, another question. Yeah, your name?
Sir, Prabhav Pradeep. Sir, from Kendra Vidyalaya number 1 Kanchan Bagh. Sir, my doubt is, you said that if we get a coronavirus, we have a unique sentence from which we can develop the CRISPR. Sir, but I have a doubt. What if we have an unknown virus X, whose sentence we don't know?
Yeah.
So how to get that sentence?
Excellent question. Okay. Yeah. So if you do not know the sentence of particular virus, okay, by this either CRISPR or RT-PCR, it cannot be detected. Okay. So what is the alternative? Alternative is sequencing. Okay. What I do not know that which virus. I want to read the genetic material ATGC. We call sequencing technology. Okay. We take DNA, RNA. Okay. And we sequence them. Once I know the sequence, I will read, and not only I read, I also try to identify what kind of sentences are there. So coronavirus when it came to our society, it could be China, it could be somewhere else. There is no RT-PCR. Nobody knows whether it is coronavirus or not. Okay, they sequence the virus, then they match to it all the known virus. Okay, and see whether they are similar or different. Okay, and they traced it is a new virus, how they evolved, what is the original virus was. Okay, so if you go back, SARS is a basically original virus, and then they have mutated and came out with a COVID-19. In 2019, it is a new strain has come, and only sequence which will allow you to identify a new virus. Okay, like reading ATGC or reading the novel from start to end and put in a kind of text format. That's the only way to do it.
I think we'll allow another student to ask question. Yeah, please.
Good morning, sir. This is Sevita from Krishnavani Talent School. I want to ask you one question, that is it still necessary to wear a mask? As we see here, only minimum members wear mask. Is there any possibility to getting corona again?
Yeah. Is it any possibility to get coronavirus? I will tell you yes. Okay. Is it going to be same coronavirus? As you heard, there are different strain, Omicron, Delta, XYZ. Okay, many names. Okay, 4.240. Okay, this is a strain. Okay, same virus with a new, kind of activities. Okay, it may come. Okay, but virologists, those who works on virus, okay, they are the master who can give you the correct answer. Now, everybody gives answer against by coronavirus, but what they tell, when the first hit comes or wave comes, there is a second, then third, then like a wave, its intensity goes down. Okay. Now, depending on the life cycle of the virus, okay, how long it will go down, it differs from virus to virus. For coronavirus, it has been predicted almost it has gone. Okay. Now, pandemic, that was the pandemic. Then when virus do not come in a wave, we call epidemic. Okay, we have reached to the epidemic. Okay, seasonally it will come because it doesn't come each and every season of a year. It is seasonal specific virus. Certain season it will come, but it will not be that dangerous as it was earlier. So this wave has gone down in a region of epidemic. We are not in pandemic anymore.
I think another 2 minutes are there. Dr. Madhav Chattopadhyay, our Jadavpur University alumni, also an eminent scientist, was of CCMP. Madhav wants to ask you a question, Dr. Souvik.
Good morning.
Morning.
Antibiotic resistance of bacteria is a global challenge. So how CRISPR could be useful in the management of the problem?
Management of
this problem, antibiotic resistance.
Antiviral resistance.
Antibiotic resistance or antibiotic resistance, how CRISPR can help us. Am I right? Okay.
So at this moment, CRISPR has not seen any application to combat with the antibiotic resistance, because it's again comes due to the change of the genetic context of any bacteria or virus or fungi. So CRISPR basically can make the change, cannot stop the change. Okay, but you never know, in near future there will be a chemistry which will be introduced in the CRISPR system, which can prevent to have the genetic change, so that bacteria will remain, same bacteria will not show any resistance. It may, but at this moment there is no such example where CRISPR has used to prevent antimicrobial resistance. Yes, sure.
All sorts of strains are coming. Whatever diagnostic test.
Yeah.
Is it for all these strains, or you have got a
specific?
It
is. So far we can detect all the strain. Not only that, we can also detect and tell that which strain it is. Okay, design is No, single will not help. That will be different. First thing, whether corona is there or not, it is easy to. We have a selected sentence which is unique for each and every one. Then we need to do. We need to pick up a sentence where the variation is coming. So there. Also, we have developed test on that.
Thank you for the wonderful questions, and thank you, Dr. Maiti, for answering them so nicely, and also for explaining the difference between invention and innovation. I think this is a wonderful indigenous invention that we have had, and some innovations do take some time to scale up, So I would now request our president and senior member, Dr. Amul Chakraborty, to felicitate the speaker.